All right, we're recording. So welcome to our discussion on PCR, polymerase chain reaction. So this is a beginner's guide kasi beginner rin ako. Okay, so let's begin. So we're going to introduce the different and important concepts, fundamentals, basics on PCR, and the different types of formats or other types of PCR. So we collected your DNA extract and now we will process it to PCR. We will amplify our target DNA and after PCR, we will run it to gel electrophoresis to see the types of genes we have amplified. Tingnan natin under gel electrophoresis. We could visualize it. And if we want to do more, okay, we could do DNA sequencing. We could send it over to Philippine Genome Center. So this is a basic setup of your PCR. So you have your containers for the pipette tips. Your reagent should supposed to be on ice. So I will get the reagents from the freezer. And we have our micropipette with our filtered tips. So, yan. So, it should be sterile. That's why I be letting you to bring a spray bottle with 70% ethanol so we could dissolve lipids and proteins and unnecessary DNA. So, this is what your PCR looks like. We have three major steps. We have denaturation, wherein there is separation of your DNA strand. In annealing, we have three important reagents. We have primers, polymerases, and your nucleotides. Your primers will be the bookmarks or site or the marker where your DNA attack polymerase will extend or elongate the new strand. So first is denaturation. So this is our template DNA or extracted DNA. At 95 degrees Celsius, the hydrogen bonds will separate. So we have your attack polymerases. It is resistant to very high temperature. Kung DNA polymerase, hindi magsisurvive yan sa 37, more than 37. So ito, yung mga primers, they are marking the spot, the area, the target gene where it will be amplified. And these nucleotides are the building blocks to create a new strand. So, ganun na. So, our PCR, I set it, our thermocycler, I set it at 35 cycles. So, ayan, nagmumultiply siya ng 35 times. So, 2 raised to 35. So, ilan kaya ang 2 exponent niya, 35. So, ilan kaya ang nabubuo ng 2 raised to 35? Yan. So PCR is a way of amplifying our target gene, our target DNA. It's like we're photocopying it multiple times. Yan. So that's PCR. So, ayun. So it's invented by Carey Mullis in 1983. So he was working in Cetus Corporation, California. And this corporation, guys, they have this product which are oligonucleotides. So nalulugi na tong Cetus Corporation and they're finding a way on how could they maximize the use of oligonucleotides so para maging mabenta siya sa market. And apparently, guys, these oligonucleotides became our primers. Yan. So, primers are designed. So, I talked to a commercial company, tapos nag-order ako ng primer. So when I was starting on PCR, I don't know, hindi ko naman expect na mamimili ka pala ng primer because you will choose a target gene that you want. And I asked them, what do other schools use as a primer? And they said 16S rRNA. So, yon, a company will design it, will find what is 16S rRNA, and they will design it for you. Yan. So, kung anong gusto mong primer, yun yung gagawin nila. So, the same thing with Cetus Corporation. Because of the invention of PCR, a lot of laboratories now are using PCR, and the patent for PCR, the patent for primer, sobrang yumaman itong Cetus Corporation California and Carey Mullis. So, so this is the start of the thermocycler. So thermocycler is the machine that is used in our PCR. Yan. So, ang ginagamit lang natin sa academic level na PCR is a conventional PCR. During the time of COVID testing, RT-PCR, yan. So, RT-PCR, yun dapat ang ginagamit. So, RT-PCR is different from conventional PCR. Yan. Kasi pag sa RT-PCR, nakita mo na yung amount of target gene that was amplified, it provides more information than compared to conventional PCR. RT-PCR, hindi mo na kailangang i-gel electrophoresis. Tayo, kailangan nating mag-gel electrophoresis. Yeah. So, in 1953, your double helix has been invented, and invention of PCR is 1983. So, tagal na, no? This is not a new technology. It has been used for a very long time already, okay? So, primers was discovered in 1971. The first DNA the DNA polymerase in the body was discovered in 1957. So, the DNA polymerase counterpart in our PCR, guys, is Staq polymerase or Thermus aquaticus. Yan. So, sobrang the discovery of this bacteria has revolutionized immensely also our PCR. So, ang halaga na, no, mahalaga yung mga primers, Staq polymerases. Kung wala yung mga yan, there will be no amplification that will happen, ha. So, if you are making your master mix reagent and you forgot to add the Staq polymerases, you forgot your primers, there will be no amplification that will happen. So, sayang ang paghihintay natin ng less than 2 hours. So, what is PCR? So, it is an amplification of specific parts of your DNA. How? By the use of enzymes, specifically your Staq polymerases, and cycling condition. How? There is a cycle of temperatures. May 95 degrees, may around 55 degrees, may 72 degrees, and so on. It's a cycle, okay? So, dati, ang hirap mag-PCR because they just use water bath and they transfer it manually, okay? Ngayon, yung mga conventional thermocycler natin is they change the temperature continuously. High-tech na sila. So, your PCR, it could be further used, processed to gene sequence. You could now, so once you amplified the gene, you could now sequence it. Malalaman mo kung ano yung genetic code na andon. So perhaps, maka-identify tayo ng bagong species ng bacteria. Next, it could be used in the clinical setting to determine mutations. Example, factor V laden mutation. So in hematology too, this factor V laden mutation is one of the most common hereditary thrombosis. So, namamana ito and you are prone to thrombosis. to clot formation pag may factor V latent mutation ka. So it could be determined through PCR. And the diagnosis of infectious diseases, new diseases, emerging diseases, re-emerging diseases, old diseases. So, could your PCR detect antigens and antibodies? Of course not, okay? It determines nucleic acids. So, the gene that codes this antigen and antibody, pwede niya pa ma-determine. But detecting antigens and antibodies, of course, it is not part of PCR. It's part of serology, okay? So, it detects the genetic materials. So, sa topic natin ng chromosomes, it could detect plasmids. It could detect viruses. Are RBC suitable for specimen for PCR? Yes or no? So the difference between replication and PCR. Yan. Dapat alam nyo na to. So sa replication, to separate the two DNA strand, we have a helicase enzyme. Outside the body, through the thermocycler, we have denaturation. In the body, we have DNA polymerase. It's very active at less than 37 degrees Celsius. On PCR, it's stuck polymerase. Yan. So, it could withstand high temperatures. Replication is a continuous process. So, it never stops because you need it in the creation of new cells, new daughter cells. For PCR, it's discontinuous. It stops. In replication, there's less error. In PCR, there's more error. Okay? And it's lower. So PCR in vitro outside the body, replication inside the body in vivo. So fundamentals, we'll talk about the components of PCR, the steps in PCR, and how to ensure a successful PCR test. All right. So, ito yung pinakita ko kanina. Kanina cycle 1 pa lang yon. So in cycle 1, there is the creation of two strands. So 2 raised to 3, 8. So 1, 2, 3, 1, 2, 3, 4, 5, 6, 7, 8. There should be 8 new strands in the third cycle. So kung ano yung cycle, yun yung exponent, no? So in 2 raised to 30th, on the 30th cycle, expect 10 raised to 9 copies of DNA. Okay? So sa first cycle, 2 copies of DNA. Sa second cycle, 4 copies, 2 raised to 2. So 2 times 2. Sa third cycle, 8 copies, 2 times 2 times 2. Okay, huwag niyong kakalimutan yun, ha? So, major steps in PCR, we have denaturation, very simple breakdown of hydrogen bonds to separate the two separate strands. So, they will be the template DNA. So, dito, dahil sa paghihiwalay nitong dalawang to, sila ay magiging basis for the new strands. Okay? So denaturation happens at very high temperature. That's the temperature where your hydrogen bonds will break. It could happen around 1 to 3 minutes. So if you will notice, when we will be using our thermocycler, there will be an initial denaturation and denaturation. Yeah. Next is annealing. So this happens cyclical, no? So annealing is very important because this is the time where your primers attaches. You have 2 primers, forward and reverse primers. So the forward primer attaches to the 3' end of one strand. Pareho sila sa 3' end nag-a-attach. So annealing temperature is computed by subtracting 5 that melting temperature minus 5. Melting temperature of what, ma'am? Primers. So, annealing is very highly dependent on primers, okay? You could not set the temperature of annealing if you don't know the melting point of your primers. So, how is annealing temperature computed? It is lowered to approximately 5 degrees below the melting temperature of the primers. Yan. So, the primers that I bought, the melting temperature is 56.1 degrees Celsius. Hence, I set the annealing temperature -5, 51.1. Yun ang annealing temp ng thermocycler natin. Kasi it depends on the melting temperature of the purchased primer, ha? Pakalimutan. So, this is the time where your primers bind to the target sequence and also your DNA polymerase. So, yung primer muna mag-attach siya, then your Taq polymerase. Alright? Not the other way around. Because primers act like a bookmark. It will mark where your DNA polymerase will start to create a new strand. Entiendes? Alright, next guys is elongation and extension. So after the addition of reverse primer, after the addition of the Taq polymerase, now the green one is the newly synthesized DNA. To be able to elongate and extend the new strand, para mapahaba mo ito, you need DNTPs, your deoxynucleotide phosphate. DNTPs. Yan, that's your assignment. Ha? What does DNTPs mean? So your DNTPs are elongates, extends your new template. They are the building blocks of the new strand. Yan. Sabi niya, bases are added to the 3' end of the primer. 3' end of the primer by the Taq polymerase. This elongates the DNA in the 5' to 3' direction. 5' to 3' direction. 5'. So the primer attaches to the 3' end. But however, the Taq polymerase extends the strand from 5' to 3' direction. Yan. So, elongation, in your notes, in your manual, elongation will depend on the number of bases of your primer. So, it's ganon. The time so the time of elongation will base on the how long is the primer. So our primer 6S rRNA is about 20 seconds. So instead of 2 minutes in your thermocycler, I made it 21 seconds at 72 degrees Celsius according to the protocol. Alright, so this is what your screen will look like in your PCR screen. Yan, you will set it, ikaw magse-set nito. So you will have initial denaturation, annealing, extension, and final extension. So if you're going to work in the laboratory, you should read the manual of your reagents. Okay? So, the time is from the manual of the reagents. Yeah. So, hindi ko lang inimbento yun, no? So, this is another depiction of how your PCR would look like. So, itong yellow, this is your Taq polymerase. And the white ones are the DNTPs that makes up the new strand in extension and elongation. So this maroon one and green one are your reverse and forward primers. Yan. So yung andito, forward primer. Yung andito, reverse primer. Okay. So, what are the components of PCR? So, very important. Okay? So, pag may nawawalang component ng PCR, isa man dito mawala, hindi tutuloy ang PCR. There will be no amplification. Okay, first one is you need your DNA template or your target DNA. Ma'am, saan ganin to? You got it from your DNA extraction method. Yan. So, it is very important that the extraction method has high purity, okay? Or else, it will create poor PCR. Tingnan natin. Because, as we all know, our DNA extraction method was very imperfect. So, let's see, how will it affect our PCR results? Yan. Abangan natin, no? So, ma'am, what are the types of template that we could use? We could use genomic DNA. That's what we use in our PCR. Genomic DNA, DNA from humans. And you could use complementary DNA. So, to get a complementary DNA is, guys, sa PCR, ang pwede mo lang i-amplify, mga DNA lang, okay? You could not amplify RNA, okay? You could not amplify RNA. So, ma'am, how would we amplify RNA? So, RNA plus reverse transcriptase equals complementary DNA. So, your RNA must be converted to complementary DNA so that you could amplify it in your PCR. Okay ba tayo? So, yun na. Sa PCR, RT-PCR, all PCRs, they only amplify DNA. So, to be able to amplify your RNA, you have to convert it into a complementary DNA. How? By adding an enzyme which is reverse transcriptase. Okay. So, your primers are the one that attaches to the target sequence. Silang nagdidikta kung ano yung i-amplify. And DNA polymerase, they are the construction workers. They are the one who will add the cement, the nucleotides. So, your reverse primer, your forward primer, dito siya sa baba, na strand. Anong strand yan? That is the antisense strand or the coding strand, okay? So, forward primer, nasa babang strand, it attaches to the antisense strand. So, ang tawag dito sa babang strand is called antisense or non-coding strand or template strand, okay? So, yung reverse primer, naka-attach naman siya sa taas na strand, no? So, it is called the sense strand or non-template strand. But, sa totoo lang, they are both used as template, no? So, names lang ito. So, what happens if you only use, yan, nakuha ko to ata sa BYJU site. Ang ganda ng site na yun. What happens if you only use a forward primer? If you only use a forward primer, only the template strand will be synthesized, but it will not have a complementary strand to bind to. Hence, no amplification will occur. So, napoprove yung point natin na pag may kulang na reagent is hindi natutuloy ang amplification. Walang nare-replicate, okay? So, does PCR use DNA or RNA primers? PCR uses DNA primers. RNA primers are used in replication in vivo. Alright. So, what are the properties of a primer? So, I told you, primers, you design it, okay? A primer has to be specifically designed according to the strands. So, depending on your need, okay? So, for our need in the laboratory, we need to amplify bacteria. So, the primer that I chose is something that will amplify a gene that is common to all bacteria. Okay? So, if a scientist needs to amplify SARS-CoV-2 virus, specifically Omicron variant, they need to get a specific primer for that. Okay? So guys, mura na actually ang magpa-design ng primer. So, ano mo na lang, imagination mo na lang ang limit, no? So, affordable bumili ng primers at magpa-design. Okay? So kayo, if you are planning to do research, so you could purchase a primer. Mag-amplify kayo ng sequence na gusto nyo. Diba? So, ma'am, how do we design a primer? So, we have software tools found online to craft your primer. After you have designed it, you will send it to a commercial company that makes primers. Yeah. Tapos yun, bibigyan ka ng tube na, na mukhang may aqueous solution. Primers ang naman. Alright. So, usually, our primers are about 18 to 24 base pairs in length. Ganun sila kahaba. So, ACT, GGT, ACC, TAC, yan. So, ganon, 18 to 24 in length. It should be specific to the DNA region that is amplified. The melting temperature for both forward and reversed primer should be similar in range. Highly repeated sequences should be avoided as it can lead to formation of loops in the primer. So guys, designing a primer is an art as well, no? Kasi pag masyadong magkakapareho yung sequence, yung magka-opposite lang talaga yung reverse at saka forward, baka sila yung magdikit sa isa't isa. Yan. Ganon. Hindi mag-attach sa target sequence, baka mabuhol nila yung isa't isa. Parang ganon ba? The primer's design should be different from each otherwise they can anneal to form dimers. Yan. Baka magsama yung, instead na mag-attach yung forward primer sa anti-sense strand, yung reverse primer sa sense strand, baka sila na yung nagdidikit. Ganun. So, it's also a art. So, this is what I brought. Sorry. This is what I bought. 16S rRNA primer. So, this is a good primer because 16S rRNA is a gene that is highly universal. It is found in bacteria, archaea, yun. Halos lahat ng bacteria. It is extremely conserved. So, ibig sabihin, majority of bacteria have 16S rRNA gene. Yan. Mapag-gram positive, mapag-gram negative. Yan. So, it is rRNA. So, it is found in the ribosomal RNA, in the small ribosomal unit of bacteria. Right. So, kung gusto nyo mag-identify ng mga new and novel bacteria, pathogenic bacteria, 16S rRNA primer is a good sequence. Okay? So, in your molecular biology, I know this is your favorite, diba? You have cultured your bacteria. Yan. So, in your culturing of bacteria, you differentiated them based on their cultural characteristics, saan sila nag-grow, tama. Tapos, ano yung biochemical identification nila, di ba? Kung ano, KSA, yung mga ganyan. If it's lactose fermenter, ganyan. At kung ano-ano pa, no? So, in microbiology, ganyan ang purpose niya. But guys, if you really want to know the taxonomic classification, if you want to really know talagang sino kamag-anak ng bacteria, the molecular classification, if you want to reclassify that bacteria, if it's really not sure kung anong classification ng bacteriang yan, what we use is gene sequencing, okay? So after PCR, bring it to Philippine Genome Center and have it sequence so that we will really identify kung ano yung bacteria na nakuha nyo. Malay nyo, yung bacteria na pala na yun is a new species and novel pathogen. Yun ang contribution ng molecular biology, all right? If you really want to be really specific on the family, on the genera, on the phenotypic characteristic of the bacteria, use molecular biology. Yan. Bilib na ako sa microbiology nun. Because wow, iba yung growth. Pwede mo may differentiate kung E. coli o Staphylococcus aureus. But if you really want to get to know that bacteria, you can forgo phylogenetic analysis. Talagang makikita mo kung sino mga kapamilya niya. Yan. Go for molecular biology. So, ayan. Ba't biglang Taq polymerase? So, yung Taq polymerase, yan, is very... So, discuss ko pala isa-isa. So, Taq polymerase, dito, what makes a good DNA polymerase and why Taq polymerase is popular? So, Thermos aquaticus, guys, is a very popular source of polymerase, no, for PCR. But we also have other bacteria used for polymerase. Meron nga tayong PFU. Could you search what bacteria produces PFU, DNA polymerase, no? So, itong mukhang, ano na to, mukhang Pacman, guys, is your polymerases. So, what makes a good DNA polymerase? They should be highly specific. Sabi niya, low non-specific amplification. Kung target gene lang, kung 16 sRNA lang ang pinapa-amplify mo, yun lang. Okay? Wala nang iba. So, it should be highly specific, which is your Taq polymerase is highly specific. It should have high thermostability. It should not be destroyed in the course of your PCR, no? Kasi ang taas-taas ng temperature ng PCR, it could reach up to 95 degrees Celsius. So, kung masisira yung polymerase mo at 95 degrees Celsius, sayang pinaghirapan mo, no? No amplification. So, it should withstand high temperature wherein kinakaya ni Taq polymerase. Good processivity. It is fast. Yan. It could add 60 bases to your new strand per second. So, it's a very hard worker siya, no? So, naglalagay siya ng mga hollow blocks, 60 hollow blocks per second. Parang ganun ba? So, average fidelity or proofreading capacity, mas may tatalo. Yung PFU, mas magaling kay Taq polymerase na mag-proofread. Yan. So, mas mababa yung error rate. Unlike your PFU is 7 times better in proofreading compared to Taq polymerase. But its synthesis rate is less than half of that Taq polymerase. Mabagal. Mabagal yung processivity ni PFU. Yeah. So, gano'n, mamimili kayo. Pero talagang walang tatalo sa Taq polymerase. Depending on your need on it. Okay. So, we have your nucleoside triphosphates, your DNTPs, deoxynucleotide nucleoside triphosphates. Yan. So, ito yung mga ina-add, no, sa DNA sequence. Yan. To extend it and elongate it. Meron tayong magnesium chloride. So, watch out, no? Kasi meron tayong manganese chloride. So, yun, magnesium chloride, it is the cofactor of your DNA polymerase. So, to improve the attachment of DNA polymerase to your strand in primer, kailangan ng magnesium chloride. Okay? And buffer. Your buffer maintains an alkaline environment. So, this is your magnesium ion. Magnesium ion attaches to your polymerase, itong green, to improve its attachment to the primer. Yan. And template DNA. Yan. Basta, it improves the attachment. helps coordinate interaction between the 3' end of a primer and the phosphate group of an incoming hollow block. Yan. So, to stabilize talaga yung structure, kailangan natin ng magnesium chloride, no? So, hindi yan extra. Kailangan talaga yung magnesium na yan. So, how could we ensure a successful PCR test, no? So, sterile environment, yan. Picturean nyo na lang, no? Basahin nyo na. Inventory of the ali-coated PCR reagent. So, freezing and thawing the reagents too many times could damage the enzymes and DNTPs due to recrystallization. So, pag nag-freeze ka, there are crystals that are formed, okay? So, your reagents, so dapat hindi sila nalabas sa ref, matigas, tapos name-melt, matigas, nagme-melt, ganyan. So, dapat stable siya or i-alikot mo siya. Okay? So, monitor your reagent na hindi masyadong labas-pasok sa freezer. Yan. So, kung mapapansin nyo sa PCR natin, I will aliquot the reagent. So that para maiwasan rin pag may natapon kayo, ganyan, hindi ubos yung lahat ng reagent natin, no? So, ensure to have a correct annealing temperature. So, annealing temperature is -5 or 3 to 5 minus your melting temperature of your primer. So, too much high, if your TA is too high, low product yield. Mababa yung mga amplicon na pag masyadong mainit. Pag too low naman, high number of base pair mismatch. Diba? So, ang mahalaga pala yung annealing temperature, hindi basta-basta. Ito. Check off each reagent as if it is added to the master mix. Make sure each reagent is added in proper concentration and is not left out. Okay? Kasi leaving out a reagent, no amplification. So, make sure mo sa master mix, ma-add mo lahat ng reagent needed. Alright? Check the DNA quality. So, your template DNA, syempre, mas maganda yung quality, mas maganda yung PCR result, okay? Kung pangit yung PCR, yung DNA quality, pangit rin yung PCR result. Magnesium concentration, mahalaga yan. So, nandito na tayo sa types of PCR. Hindi na lang, matatapos na tayo. So, we have 4. Yan, 4 dapat to. Multiplex. Okay, madali lang to. So, what we have in the school is conventional PCR. But in the hospitals, they have real-time or quantitative PCR. Yan. So, it has high resolution. It shows graph or peaks. Unlike our conventional PCR, we need to run it on gel electrophoresis pa. So, mas maraming steps pa, no? So, at least may graph and peak. So, like millions kasi ang RT-PCR, kaya wala tayong PCR. So, real-time PCR ha, or quantitative PCR. Yan. So, the keyword is, it has a fluorescent reporter. Yan. So, that is what makes it special. So, this fluorescence probe or reporter, the signal strength of the fluorescent report is directly proportional to the number of amplified DNA molecules. It will really tell you how many molecules were amplified. Okay? So, just like in your COVID SARS-2, okay? By 25th cycle, kung mataas na yung amplification, positive ka sa SARS-CoV-2. Okay? Pero by the 20th cycle at mababa pa, hindi pa na-reach yung threshold, wala kang negative ka for SARS-CoV-2. So, may iba po yung real-time PCR sa reverse transcription PCR. Okay? So, it is good for RNA no? So RNA plus reverse transcriptase, it will form a complementary DNA. So yun, ang makakabasa nun is an RT-PCR machine. We have nested PCR. This was designed to improve the sensitivity and specificity. So if you want you want a more specific amplification, kung magne-level up ang UV, dun tayo sa nested PCR. Okay? It will reduce the non-specific binding. So yung mga non-specific na amplicons, mababawasan. Fourth, guys, is multiplex PCR. Sa normal na PCR, isang sample, isang primer lang. Ayan o. Isang tube, isang primer, isang sample. Sa multiplex, guys, multiple samples, multiple primers in one tube. O, diba? Ang bongga. So, yan ang multiplex PCR. You could amplify more than one sample, more than one primer in one run, in one tube. Unlike your PCR. That one sample, one primer set lang. Tag-isa lang per tube. Yun, multiplex. Ganda, no? So, this is where our PCR ends. Thank you guys for being with me. Bye-bye!